Yeah, so I'm going to talk about dogs. Caveat to begin with, these are not dogs. These are cats. These are my cats, Darwin and Lacey, who's named after Ada Lovelace. I got into dog genetics because they're fantastic genetic models. It turns out they're pretty cool animals as well, but it's been really interesting learning all about them. This on the left is a wolf, and that on the right is a dog. They're both giving us saliva samples, which is very nice of them. Dogs are really interesting because they're actually a subspecies of wolf with some really key differences in their social behavior, such as things like sensory integration, their ability to socialize with other species, their ability to generalize in their environment and things like that, and their likelihood of eating you being an awful lot lower. And this, as I said, they're actually not that far apart. So dogs were domesticated from wolves probably somewhere between 15 and 30,000 years ago. Right now, this is suggested to have happened in Europe or Africa or the Middle East or Central Asia or East Asia. And I was at a meeting a couple days ago where the people that work on this then proceeded to get into a big argument about whether domestication could be considered an event. So stuff happened a long time ago. And then a lot of time passed. And at this point in time, dogs are probably mostly just village dogs that kind of hang out with us, live in our environment. They're possibly helping us with things. Dog breeds, which is what people really tend to focus on in terms of dog genetics, are actually much more recent. So the vast majority of dog breeds probably came up within the last 100 to 200 years. And that's when we were doing really strong selection on things like what they looked like. Each breed has very limited genetic diversity because they're all really related to one another. And this means that often they've kind of like captured certain diseases at very high rates. You know, if one of the dogs that was used to found the disease had cancer, because there's such limited diversity going into it, it then becomes a very common disease in the whole breed. But there's also really interesting behavior going on with the breeds, and this is kind of what inspired us to start looking at behavior in dogs. How do you actually end up having these variations in behavior between the populations? I mean, anybody that's ever owned, you know, a dog that likes to retrieve balls, like a golden retriever or something like that, if they happen to have the retrieving, they can do it for hours and hours and hours on end. A lot of people that have dogs will know that their dogs are not the least bit interested in retrieving balls. There's an incredible amount of variation. And what we've kind of been trying to ask with dogs is, how do changes in DNA actually lead to changes in something as complicated as behavior? And because we're working in a natural system, can this actually help us understand how adaptive behavioral traits evolve over time? And we're doing this through a citizen science project we set up called Darwin's Dogs. It's a collaboration between the University of Massachusetts Medical School, where I have a faculty position, the Broad Institute, and LAABC, which is actually an organization of things called animal behavior consultants. So these are people that are like dog trainers, so the people that are actually out there working with people and their dogs. So I'm first going to take you back into the mists of genomic time, 2005, which is when we actually sequenced the dog genome. This was the fourth genome that was actually completely sequenced. We had a lot of fun with the nature cover, and actually we got permission to Photoshop in Tasha, the dog we sequenced, into the photo. Just like the dog genome is very similar to the human genome, as was mentioned earlier. There's a lot of similarities between species, especially between mammalian species. We sequenced Tasha, a female boxer. It annoys everybody that works on Y chromosomes because it means we don't get Y. 2.4 billion bases. The human genome is about 3 billion, and about the same number of genes in humans. Pretty much they share the kind of same common gene set. And as with human genetics, we're left with the same question, which is, now we've got a genome sequence. We have no idea what it does. And as with humans, one of the biggest challenges we're dealing with is we've got these billions and billions of bases in the genome. Only about 1.5% of that is actually coding for genes. And probably another 7 to 10% of it, the sequence of the DNA, is actually important in terms of the actual organism itself. And so actually figuring out what parts of the genome are actually doing something is still a huge challenge. Just like has been mentioned in many, many other talks these days, we're doing basically your genome-wide association study to do this in dogs. You take a bunch of dogs that have the trait, you take a bunch of dogs that don't have the trait, you compare them, and you look for places in their DNA that are different. And to do this, you just get a whole bunch of dogs, get a DNA sample, run it on a fancy machine, and get a whole lot of data out. And at that point in time, we take each SNP, which is basically a marker in the genome, and we ask, at this position, are these two groups of dogs, similar to each other? We compare them all, and we basically look for the position where, all of the, if this was a single gene trait, all of the dogs that had the trait were different from the dogs that didn't have the trait. Simple introduction to GWAS, which would have been far more useful if you guys hadn't already heard about it all day. So When we started this, we started working in dog breeds. We were looking at coat color and boxers. This was the first gene that we tried to map in dogs. We took nine solid boxers, so these are boxers that didn't have any white fur on them, and 10 white boxers and compared them all. And this is an interesting trait because about 2% of the white boxers are deaf, which actually comes in later on. We did our genome-wide association study. You're basically looking here at the position in the dog genome. Dogs actually have 39 chromosomes, far more than humans. That's why there's so many different colors on here. And at each position, we asked how different they are. We got a fantastically significant association on chromosome 20 of about 9 times 10 to the minus 8, and that was sitting over a gene called MID-F. Great, worked beautifully. We looked at what MID-F did, and this is where it got interesting. So people had looked at MID-F before in terms of pigmentation because it was linked to several pigmentation-related diseases in humans, but they hadn't been able to find any mutations in the coding regions, the regions of this gene that actually made the MID-F protein. And when we looked at it, it turns out that it comes in 2 different forms. There's an A version, that's the version that you need. If you don't have the healthy A version, if you don't start making your gene there, you have bad eyes. But if you actually have to start much further down the gene where that M is in order to make the one that you have to have for normal pigmentation. And when we sequenced our dogs, what we found was that they actually have mutations not in the coding portions of the gene, but in the regulatory portions that controls probably when and how much of that M version of the gene that's being made. And this is why these dogs all have totally fine eyes, because we haven't changed the coding portion of the gene. We've only changed how much we've regulated how much of that M version is getting made. And actually, the really cool thing about this is if you actually look at the dogs, it totally makes sense. So during development, what happens is that pigment cells actually arise along the neural crest, where your spine is, and they basically spread across your body. And what we have here is a mutation that's impacting pigment cell migration. So the solid dogs have pigment everywhere. The dogs that have one normal copy and one mutated copy of the gene have half pigmentation, so they have a white chest because the pigment cells don't make it all the way around. And as it turns out, you have to have pigment cells in your ears in order to be able to hear. So the dogs that have no pigment cell migration at all actually end up being deaf. And that's only about 2% of the white dogs, but it is there. And actually, if you look at the white dogs, they'll often have a big patch of color on their head, and that's one pigment cell that's managed to migrate in the early stages of development. And I think this really does illustrate the power of dog genetics. They'd studied this gene many times in mice before, and they'd broken it to see what it did. And it turns out that if you break this protein, you get mice that are white, but they're also deaf and blind and mostly dead. So it wasn't super informative. With dogs, we wouldn't have accepted a mutation that killed the dog. We needed to have something that was much more subtle, that just kind of changed the way the genome was used. And so instead, we have a regulatory variant that makes these dogs white, but they're very rarely deaf. They have totally normal eyes, and critically, they are not dead. And so this was great, and we went on and we studied a whole lot of these kind of single gene traits within these kind of inbred dog breeds. There's an ALS model, there was the Ridge and the Ridgeback and things like that. And we found a bunch of them, and it seemed to be working really well. But then we moved on to more complex traits. And so this is actually compulsive disorder in dogs, which is thought to be a model for human OCD. It's basically defined, OCD is defined as doing an otherwise normal behavior so much that it disrupts your quality of life. And so dogs obviously are doing dog-related behaviors like spinning, chewing on themselves, snapping at things in the air, chasing shadows on the floor. It's distressing, it's time-consuming. They have a very similar age of onset to humans. In both species, it's a highly heritable disease. They're actually treated with exactly the same drugs, SSRIs like Prozac and some other drugs, and they have almost exactly the same response rate to the drugs. So about 50% of dogs and 50% of people are getting some kind of relief from these drugs. And the fact that they have a similar kind of response to the drugs actually may suggest that we're looking at similar pathways in the brain. So we did a study a few years ago. We compared 92 Dobermans with OCD to 67 healthy Dobermans. You'll notice this is a much smaller number than we have in our human studies. That is because of the limited diversity in these breed populations. We got a really nice peak. It was over a gene, which is always good, because when you hit a region without any genes, it's super annoying. Only one gene. It was a gene called CDH2, and as it turns out, CDH2 is a neural cadherin. It's basically involved in linking the presynaptic and the postsynaptic membranes. And it's found in these things called glutamatergic synapses. It's involved in learning and memory. And it actually linked in really nice to a whole lot of genes that were coming out of the human genetic studies, even though those weren't reaching genome-wide significance yet. But we also discovered a problem in working in dog breeds. And so the thing about dog breeds is they're super inbred. So there's lots of regions of the genome where they don't have any variation in their population. And the second largest region of the genome in the Dobermans like this actually had a gene in it called CTNNA2. And when we looked into it, it turns out that this is actually a protein that's involved in anchoring those neural cadherins to the presynaptic and the postsynaptic membrane. And when we went on and later did a study that involved 3 additional dog breeds, we found that in those 3 breeds, we actually did see more variation in this gene in the cases than what we were seeing in the controls. And so it does actually appear to be involved in these diseases. And it may be that in some cases, and we saw this with bone cancer as well, that the way you're getting to having such high rates of these diseases in these breed populations is that essentially every single dog in the population is already at risk genetically. We went on later on and actually did a study in humans. We did a bunch of sequencing of thousands and thousands of humans. This paper came out fairly recently. We leveraged some really, really big data sets that are now available for human disease studies to actually find what we think might be one of the first genes that has been significantly associated with OCD in humans, this orexin-1. But what we actually found was, when we set up this study, we were like, we're going to test the dog genes and see if they're involved in humans. And what we actually did is we tested those dog genes and a whole lot of other genes that acted in the same pathways. And what we found is that it's not necessarily the same genes that are involved, but it seems to be genes that are acting in similar shared pathways. And so this figure we made just shows that I think we've got the, so the human genes, the genes that have been found in human studies are labeled with little human heads. The ones that have been found in mouse studies are labeled with little mouse heads, and then the dog ones have a little dog head next to them. And what you're seeing is, first of all, they do seem to have some functional interactions with one another, but also that there are a lot of genes on this picture. Behavioral traits are complex traits. And if we actually go back to that original OCD study in the Dobermans, what you see is that even though we did get a nice peak, we're still only explaining somewhere between 5 and 10% of the risk, even within that one breed population. So there's a lot more going on. And this is kind of where we stood back and said, okay, how are we actually going to get to the point where we have enough dogs that we can actually study these complex traits? And at the same time, do we really actually need to use these weird breed populations? And so if we go back to that slide I showed you at the beginning with the dog breed history, you'll remember we have domestication, and then we have 15 to 30,000 years without any dog breeds. And this is a point in time when we've got a lot of selection going on behavioral traits in dogs. Humans are settling in permanent settlements. They've got agriculture, invention of the wheel. All of this stuff is changing the human's environment, so it's also changing the dog's environment. And if you actually look at genes that are different between dogs and wolves, a lot of them have to do with things like carbohydrate processing, basically their diet. And then much more recently did these dog breeds come up. And when we see these behaviors in the dog breeds, we're probably just capturing things that existed at a higher rate. We're not creating new behaviors within that time span. And the nice thing about these being positively selected traits is that when we study diseases, we're usually looking at things that are bad. And the thing about diseases that are bad is that either you're going to find mutations that don't have a very big effect on your risk of disease, or you're going to find mutations that are very rare in your population. With positively selected traits, you're actually looking for something that's beneficial. You're looking for something that has a big effect on your phenotype, but has also been pushed up to high frequency in your population. So you should have a lot more power to map these genes. At the same time, if you actually look at the human studies, by the time they get to statistical significance for something like schizophrenia, they have like 150,000 people in their studies. So even if it's going to be a lot easier in dogs, we're going to need a lot of dogs to do this. Luckily, there are a lot of dogs. Turns out that even in the US, there's 60 million dogs. So we launched this citizen science project. It's called Darwin's Dogs. And you basically go to our website, you sign up your dog, you answer lots and lots of questions about your dog's behavior, and then we mail you this fancy kit that looks like this, that has a saliva swab in it, and you send it back to us, and we get DNA. The questions look like this. We actually insert the dog's name into the question. This actually makes it a little bit more personal, and also accounts for the fact that if people have multiple dogs, they're not going to forget which one they're actually answering for at any given time. Things like, do they like playing with toys? If you look at the data that we have out, most of the dogs that are in our study actually do like playing with toys. And then, as I said, we send out the kit, we get the swab back, and actually a lot of times people send pictures with the kit, so now we've got a whole wall of dogs in our lab. It's quite cool. And the thing that we've actually kind of discovered as we've gone along, since we launched this about 2 years ago, is that people really like talking about their dogs. So at this point in time, we have about nearly 15,000 dogs signed up, and their owners have answered about 1.7 million questions for us. We've got about 4,000 DNA samples. I miscalibrated a little bit as to how much people really, really like talking about their dogs, so most of those 4,000 DNA samples are sitting in an office right now because I need to figure out how to actually fund the sequencing of them. I wasn't expecting it to grow quite this quickly, but it's a fantastic problem to have. And they're coming from all over the US. We actually do sign up dogs in other countries as well. We just haven't quite figured out how to get the kits out to them over there yet. And the nice thing about this, and this has kind of come up in a few of the studies here, is that actually by looking at the dogs rather than at people, even if we have a demographic skew in the people that are signing up for the project, we don't actually seem to get that in the dogs. The kinds of dogs we're getting are very reflective of the proportions of these dogs in the US population. Purebred, shelter dogs, rescue dogs, and other things. That's Nellie, who objects to wearing a giraffe hat. And we ask questions like, does your dog enjoy life? Most of our dogs are very, very happy. And does your dog, okay, admittedly, that question was actually one that was added by my postdoc because we had a 9-question survey and we needed a 10th question. And we get more comments on that question than I think just about anything else. People either love it or they hate it. And it may tell you more about the humans than it does about the dogs. We ask about basic morphology traits. This is kind of partly a positive control. We know some of the genes that are involved in these traits. We want to see if we can find them, but we also figure we might be able to find new things. We ask about those artificially selected behaviors where I feel like we might actually have a lot of power to actually map things. So things like retrieving, pointing, and even does your dog seek companionship from people? There's kind of an idea that with lap dogs, they've actually selected on that need to be in contact with people. Complex traits and disorders, there's a lot of published surveys for actually getting at traits like impulsivity, anxiety, and even personality in dogs. And we've taken those published surveys and added them to the website to get really nice kind of large sample sizes for things like impulsivity, anxiety, noise phobia, and things like that. This is a fantastic set of traits. So we sat down with those dog trainers from LAABC and we said, okay, you guys spend tons of time with dogs. We're looking for traits that show some indication of segregating by breed, because that would indicate that genetics was involved, that are easy for owners to identify and that people are unlikely to care about. So they're not going to try to train their dog to do it or train their dog not to do it. I have no idea if these traits are genetic, and if they are genetic, how they actually get controlled. But I think it's going to be really interesting to find out. So it's things like, does your dog cross their front paws when they sit down? Do they lick their bowl after finishing their food? Do they eat grass? People love that question for some reason. They're like, oh my God, my dog totally eats grass. I've always wondered why they did that. But I think the other thing you find out from asking these kinds of questions is that people really know their dogs. So it's not like they have to pause and hesitate and think about it. They know these things, and they're just kind of waiting for people to ask them. And actually, just recently, we just added on a whole set of new surveys that are actually around food allergies, because we're working with a group at the Broad that's a food allergy studying group, and also food consumption and things like that. And what we actually found, we haven't actually looked at this genetically yet, but the really nice thing is we did go back and add on these surveys, and we've actually had a pretty good rate of people coming back to fill them out for us, despite the fact that we're not actually giving them a whole lot of information back yet. And this is the most recent thing. This actually makes me very happy personally. So we have genetic information now for 374 dogs that we have survey data from. We've genotyped them on this new Affymetrix, which actually they changed companies. I think there's something else now. I can't remember right now. It used to be Affymetrix Axiom Array, which has about 800,000 SNPs on it. And we studied the trait of size. So we tried originally mailing little tape measures out to people. That totally didn't work at all. People aren't very good at measuring things, although I think there's some flaws with the tape measure as well. So instead, we just started asking this question of how high is your dog when they stand next to a normal adult person? Are they ankle high, calf high, knee high, thigh high, or hip high? And we have those 5 measures. And so we ran that GWAS, and it came out beautifully with those 400 dogs. And we're capturing most of the major size loci that have been reported in dogs, and also capturing a number of loci that haven't been reported before, including one, that first one that was actually been connected to size in horses. And so this is a really nice validation for me that we are getting good information on the morphology of these dogs, we're getting good information on the genetics of these dogs, and that this whole citizen science thing that I've been talking about for 2 years might actually work. So that's very cool. But of course, why are people actually coming to this site to study what we're doing? And of course, what they really want to know is, what is their dog? Where do they come from? They want to know something about them. And when we looked into this question, it turned out that nobody had ever actually done whole genome sequencing, so collecting all of the genetic data from a mutt from the US. People have looked at village dogs, they've looked at purebred dogs, but they never looked at mutts. And so we did this project that involved 21 dogs. The biggest criteria so basically the way you got into the study was you had to have a dog that you had no idea what it was, and you had to run into me in this 2-week period when I was looking for dogs. That was the sole criteria. It may happen to include my sister's dogs, but total coincidence there. And so what we're looking for here is, and I put this in, and this may be self-evident to some people in this room, but I just wanted to go through it. And so when we're looking at these mutts, they're basically taking these very inbred populations. There's like, I think, up to 400 different breed populations that have been kind of named across the world. And you're basically, if you think about it, if you have a dog that's a mix of 2 breeds, they get 1 chromosome from each of those populations. And then you go on to another generation, and it's not just that they get 1 chromosome from each of those populations, but you have this thing in between where you have a recombination. So you get a block switch. And so then when you make a puppy in that generation, there's got that one piece where the 2 pieces of ancestry have gotten swapped around and you have a break. You make another puppy and the same thing happens in a slightly different place. And as you go on in generations, you're going to have one of these recombination rates, basically one recombination per chromosome per generation on average. And so what you end up with in the mutts is this kind of mosaic of ancestries from different breed populations, and the more generations of mixing you have, the shorter these blocks are going to get. And this is exactly what we found when we looked at those 21 dogs we sequenced. If you look at the purebred dogs, so this is basically total homozygosity, tells you how much of the genome is completely fixed with no variation in that dog, so the 2 chromosomes look the same. And then if you look at the size of those regions, that's kind of a proxy for how big those blocks of mixing are. And if you look at our Mendel's mutts data set, because we had to call it Mendel's mutts because Darwin's dogs, you actually see that they're much, much shorter than the purebred dogs, which is exactly what you'd expect. But what I hadn't actually expected and what we see is that they're actually on par with the village dogs. And village dogs are typically defined as a dog that's kind of a dog that lives in a community with people, but doesn't have necessarily any kind of breed ancestry. They've never been through a breed bottleneck, as we say. And what we're seeing is the Mendel's mutts seem to be almost as diverse or more diverse than these village dogs. And that brings into the question of how these breed tests work. And so there's kind of 2 key things that come up when we're doing these. There's the number of markers you have in the genome, and there's the number of breeds you have to compare to. So obviously, even if you have an amazingly beautiful mutt, which has a whole bunch of really interesting things going on, if you're not testing very many positions, you're not going to see that. And you're only going to get a couple of breeds that come out as actually being tested positive for. And then, of course, if you happen to be missing, say, the purple breed in your reference panel, so you can't actually see that the purple breed exists, then you come out looking, all like the yellow breed. But what we've actually found as we've tried to get into figuring out what these mutts are is that these 2 things actually relate to one another in the sense that the more breeds you have in your reference panel, the more markers you need to actually be able to identify a breed, because you just don't have enough information otherwise to distinguish 93 different possible reference populations, which is what we've managed to get up to so far. And unfortunately, this means that given the data sets that we have right now, the reference data, we end up with a big chunk that we just can't assign to any particular ancestry in these really mixed up dogs. We don't know whether these are breeds that we just can't match to because we have insufficient information. It could be breeds we don't have in our reference panel. But the really intriguing thing is it could also just be not a breed. I mean, there were dogs in this country before, the European breeds showed up. There are village dogs all over the world. There's nothing to say that mutts have to be only mixes of different breeds. And so we've developed a breed test now. We've got 93 breeds with 12 dogs per breed. The real limitation is that we're working with the publicly available data for these breeds, which means that we only have about 150,000 markers per dog. I'd really like to have a lot more than that. But we are representing about 95% of the US breed population with those 93 breeds. And then we have a test set of about 283 dogs. And Linda, who's a postdoc in my group, although she was in my group and then she ran off to Stanford, so now I have a big issue with the Bay Area because, stole Linda. But anyway, so we have reference data from 93 breeds, and what we do is we take those breeds and we make mutts. We mix them together generation after generation after generation and make mutts. And because we've made the mutts, we can actually then develop a breed test and test how well we can actually figure out what those mutts are, since we actually know what the answer is. And that's how we actually generated this test to figure out how well it works. And then we compare it with the known ancestry. And what we found is that we're really good at single breeds. I mean, if you've got a beagle, we can definitely tell you that's a beagle. We have a little bit of ancestry coming in from other breeds, but they tend to be breeds like the bloodhound, which might actually share some ancestry with the beagles. If you have a golden doodle, golden doodle, like Hubble here, we can figure that out. We can tell you he's half golden retriever and half poodle. And probably the gray in these dogs is what I was kind of speculating, which is just that we don't have enough information to make those assignments accurately. But this is what our mutts look like. And it's actually a really amazing thing. So Skylar was actually Linda's dog. She was adopted in California from a shelter where Linda was told that she was a Labrador Pit Bull mix. Turns out Skylar does in fact have both Labrador and Staffordshire Bull Terrier in her, but her top breeds are actually Dalmatian, Rottweiler, and Golden Retriever, which is really kind of a fun thing of genetics, because once you start mixing together all of these different breed backgrounds, you end up with traits and phenotypes that people have a really hard time assigning back to particular things. And we see this with every single mutt. Every one of them is a combination of a whole bunch of different breeds. And so it really makes you question when this whole thing where people look at a dog and they say, well, clearly it's a mix of X and Y. How often are they actually getting that right? But the other thing we can actually do is look at where this ancestry sits. So this is Lucky. Lucky belongs to Jeremy in my group. Jeremy is extremely upset that Lucky's top breed is a Shih Tzu. He swears that his dog is not a Shih Tzu, but we tell him the genetics says that, so you can't argue with genetics. But if you actually look at the genome, what you can do is you can go across each chromosome and actually paint where the ancestry is coming from. And then you can actually go back and look, and you can see see where Lucky's phenotypes are coming from. So if you look on chromosome 10, Lucky's actually has this dropped ear mutation that's coming in from the toy poodles, and that's probably why he's got these lovely hanging down ears. He also carries the EPAS1 high altitude adaptation that comes in from the Lasso-Apso ancestry, so he has that mutation sitting on the haplotype that's coming in from the Lasso-Apso. Lucky, I don't think, has ever been to high altitude, so there's no way we would know this. He has the furnishings thing, which is a kind of mustachy thing that you get on some dogs. That's this gene there. It's coming in from the Lasso-Apso as well. And then he does have this one copy of this brachycephaly-linked mutation that comes in from the Boston Terriers. He has a slightly shorter snout. It's not super short. So it'll be interesting to see whether there's other genes that are contributing to that trait that actually governs how it manifests itself. A lot of the work that's been done in genetics in dogs is really focused on the power of working in breeds and ignored the major shortcoming, which is that just because you find a breed in a dog, when you find a gene in a single dog breed, you're looking at how that gene functions on the background of that breed. You don't actually get into the complexity of what happens when you start putting on a lot of different genetic backgrounds. And then we get back to that last question. The wonderful thing about citizen science genetics is that you can engage everybody in science, which, as a scientist, is an awful lot of fun. And so we've decided we're going to do this thing. We're going to take all those dogs. We actually figured out what breeds they are. And we're going to have an online contest thing where we're going to put up pictures and videos of the dogs. And we're going to ask people to guess what they think the top 3 breeds are in that dog. And we're going to see how close they actually get to what we found with the genetics. And then, of course, we're going to go on, and sorry, this is a replication. Clearly, I made a new slide the other night. And then the last thing that I kind of wanted to emphasize with this is that we've seen kind of over and over again in the talks all day that what really matters in terms of studying complex trait genetics is how many individuals you can get into your studies. We need thousands, tens of thousands, hundreds of thousands of individuals to actually really start figuring out what's going on. And we need them to be combined with detailed phenotype information. So this has to be a two-way conversation. In dog genetics, it has to be a two-way conversation with the dog owners, because that's the real power here, is that every single one of these dogs is living with some human that actually knows so much about them. And so we've really set this up as to be an open resource so that we don't actually own your dog's data. We're going to make it a resource that any scientist can get at it and look at it, and that we can share it back with the owners as well. I work at half-time at the Broad Institute. There's a whole lot of brilliant data analysts, big data kind of people there. And if you dumped a huge data set somewhere and let them have at it, they would have a blast. And it would be really interesting to see what they found. And of course, the nice thing with dogs is you can share all of the genetic data, because the confidentiality kind of limitations that come into play with human data doesn't actually affect as much with the dog stuff. And I just wanted to thank everybody that was involved in this at UMass Broad, especially IABC, the animal behavior group that we work with. They've been absolutely wonderful. And of course, all the citizen scientists that sign up and their dogs, and all the information they've given us. So thank you very much.
We have time for a few questions.
mechanistic differences are in their brains.
The mechanistic differences in their brains. So would I. That would be fantastic. No, we've actually, on this theory we have where we just get samples with the idea we will do really cool stuff with them one day. We have a project that we're doing right now with NIH to do a kind of ENCODE for dogs on a smaller scale where we get a bunch of different tissues from dogs and look at, do a TAC-seq and RNA-seq and stuff to try and get some markers of functional markers in those different tissues. But we're actually working with the Tufts Vet School right now to set up a program that would allow us to get a lot more brains from dogs. As part of, they actually have a cadaver donation program. So when a dog is euthanized there, we could actually enroll it in research and actually get the brain stored in a way that we could actually go back and start looking at functional questions after we actually find things we're interested in.
Question there?
Yeah, this is not an apocryphal question, but have you thought about doing genetics on the owners?
Yes, but IRB, I mean, it's so difficult. No.
The way you describe them, they all sound like a bunch of nutcases.
I think it actually would be a really interesting question to look at the humans. And it would be we would have to figure out how we would add it, but obviously one of the really interesting things with the dogs is kind of the shared environment question as well. And could we actually make some connections both on the human genetics, but also in terms of the human environment, education. I mean, are we actually seeing differences in environments that we could actually tie to particular health outcomes in the dogs as well? I mean, if the dogs are living in a household with somebody that smokes, they're far more likely to get cancer. We know that. And what other things could we be actually looking at if we got more information?
My second question was, if you wouldn't mind just commenting on, is I understand that in China they're doing a lot of genome engineering on dogs, and I was wondering what you thought about what they might learn from doing that.
Genome engineering is kind of the big next thing. My personal take on it right now is that in terms of actually understanding how things function, it's kind of a crude way to go about things because you're just inserting it on the level of the organism. And I think in terms of actually getting it function, what would be much more interesting would be to kind of use the genetic engineering tools, but much more on they have these cool systems now where you can grow they actually call them brain organoids, and they grow them in bioreactors, which sounds like something straight out of science fiction to me, but it's actually really cool. So you get these little systems that have all the different brain cell types in them, but you can actually get a lot more detailed understanding, once again, as you were saying, at the mechanistic level. What is actually changing in terms of signaling and things like that? I think the things that they've kind of done with the genetic engineering in China is more of these single gene traits, where you put in the mutation for big muscles, and yes, they have big muscles, but it's not entirely clear what you learn in terms of the biology with something like that.
I think we had another question. Or we did at one time. Thank you very much.
You're welcome.
We're going to take a short break.